Okay, gang, we're going to get started now. Welcome, everybody. My name is Chris Dallin, and I'm the Director of Marketing for Asep Medical Holdings, Inc. I'll be your host for today's webinar. Thank you so much for joining us today. This is our second webinar, I believe a great way for our shareholders and prospects alike to get to know us better and understand the technology that's driving our company's success. Personally, I believe that Asep is a company with genuine merit. It's based on a vision of dedication to addressing the global issue of antibiotic failure by developing novel solutions for significant unmet medical needs in human medicine, both in diagnostics and in therapeutics. And we as shareholders have the good fortune of benefiting from the innovation, hard work, dedication of our scientific team, specifically our founder, chairman, and chairman of the board and CEO, Dr. Bob Hancock. Bob is a newly installed fellow of the National Academy of Inventors in the US. This is one of the top 250 of all cited scientists worldwide, the head of the Hancock Lab at the University of British Columbia, and owner of more than 80 patents, not to mention the fact that he's a member or an officer of the Order of Canada. He's also just an all-around great guy. Also here with us today, Dr. Evan Haney, our Chief Scientific Officer, has played a key role in developing our patented diagnostic and therapeutic technologies. Evan has just returned from Tuscany, where he made a very successful presentation at the Gordon Research Conference on antimicrobial peptides. The Gordon Research Conference is the leading antimicrobial scientific conference in the world. Mr. Tim Murphy, our Chief Operating Officer, is here today, is with us as well. Tim is an experienced business executive and international lawyer. We also are happy to have our Chief Financial Officer here, Ms. Jacqueline Tucker. A few housekeeping items. This webinar is scheduled to run 1 hour. We are recording this webinar, and I will send a link of the recording to all attendees within 24 hours. There will be a Q&A session via chat at the end of the presentation, which I will moderate. So send your chats to me and I will pose the question. And we've got some very complicated science here to address. So without further ado, please welcome Dr. Bob Hancock. I think you're on, are you on mute?
Sorry about that. Welcome to my presentation. I'm going to introduce Asep in broad brush strokes, and then Evan is going to talk about the science specifically, and Tim Murphy is going to talk about the company philosophy and the progress that we've made in partnering. So we're dealing with an issue of antibiotic failure. And just to illustrate what this is, most companies are addressing this issue of antimicrobial resistance. And it's a very big one because it results in the direct deaths of 1.27 million people annually, and with 5 million associated deaths. However, it's not by any means the entire picture. So usually companies addressing antimicrobial resistance directly are trying to make new antibiotics. And that's very, very tough because there's a lot of companies in this game. Asep addresses other areas in which there's significant antibiotic failure, including biofilm-associated infections, sepsis, and situations where basically people are immune compromised. So basically all of these lead to antibiotic failure. It's situations in which antibiotics fail. As an example, I told you 1.27 million people dying of antimicrobial resistance, but 11 million people die of sepsis. So there are various approaches that one can take, including rapid diagnosis of infections to allow very specific and early therapies, and also novel therapeutics. So these strategies are being adopted by our company, and we've come up with a collection of rather diverse and exciting technologies. So we're a rare combination of both the novel diagnostic and a therapeutic approach to urgent multi-billion-dollar problems. So Asep started off life as two individual companies that were built as virtual companies. One developing a diagnostic, which is a novel assay for for diagnosis, allowing targeted treatment of sepsis, and therapeutic, which targets often incurable biofilm infections. And our overall purpose is to mitigate the global crisis of antibiotic failure and by improving patients' odds of survival and quality of life. And I'll try to convince you today that these are addressing really huge unmet medical needs. So we're in an area that in which competition is not too tremendous. So firstly, the unmet medical need perspective. The crisis of antibiotic failure is widespread and deadly. On the one side, which is the kind of yellow side, if you like, or the diagnostic area that we're dealing with, there are 49 million cases every year of sepsis. That was revealed by a Lancet article just a year and a bit ago. And 11 million people die. That's 20% of all deaths on the planet. So you might have heard of sepsis, but perhaps you didn't realize it was so incredibly deadly. And since the time that these figures came up, of course, sepsis became even more important because people who die of severe COVID-19 infection die from sepsis. So these figures, 11 million sepsis deaths per year, really pale the significance of antimicrobial resistance, which is 1.27 million deaths a year, and an area in which there's massive competition. So in the therapeutic area, we also have found a really important niche, and that is a type of infection called a biofilm, which I'll explain in a minute. And basically, we know that biofilms are super resistant to most antibiotics, and there are absolutely no drugs approved for biofilms. So usually combinations of antibiotics are used and success is not massively good. We're talking multi-billion-dollar opportunities. The global antibiotic market that tries to address biofilm infections, for example, is $41.9 billion a year, increasing by 3% annually. And the global sepsis diagnostic market is about 8.5%, but the global sepsis market overall is in the range of $70 to $80 billion a year. So the most important thing when you're starting to address a new area is to ensure that you have the backing in terms of protection of your intellectual property. And it's been said that patents are the lifeblood of biotechnology. Without it, you cannot set up a company. So on the diagnostic side, we have 2 patents families, and we've already had 16 patents awarded throughout Europe, China, Hong Kong, and Australia. And we have a second patent that is starting to diagnose the underlying causes of sepsis to try to develop goal-oriented, personalized therapies for sepsis. And then in the therapeutic area, we have 2 patent families with 18 filed patents and 8 awarded. There are 2 patents doing quite well. And we've just disclosed a new patent, which has some new peptides that Dr. Haney will talk about in a minute. So regarding the diagnostic technology, we have a rapid diagnostic test. It's built on an understanding of the underlying immune dysfunction that accompanies sepsis. So sepsis is an immune dysfunction disease, one in which your immune system starts to fight you instead of fighting infection. And this rapid diagnostic test delivers results in about 1.5 hours. And the purpose of this is to allow physicians to make better informed treatment decisions and thus to improve sepsis survival rates. So how it works is that every hour in the clinic counts. So typically a patient will present to the ER with illness. At this stage, basically, the symptoms are rather non-specific and could be any old disease. But however, the clinician, based on their experience, suspects sepsis, And from our experience, about 30 to 50% of the time, they're correct in diagnosing sepsis. So the clinician will collect blood, and then an average 13 hours later, there will be a definitive diagnosis, and they will start on particular goal-oriented therapies, especially antibiotics. However, for every hour that there's a delay in diagnosis, there's a 7.6% increased risk of death. And so because symptoms are non-specific early on and treatment is delayed, we end up with poor patient outcomes. The patient ends up in the ICU where costs are very large and death rate is 22%. So what we've come up with is a molecular diagnostic that uses a common technology available in hospitals called polymerase chain reaction. It delivers a diagnosis in about an hour and a half. This allows you to initiate immediate and specific treatment regimens, and this will improve patient outcomes, shorter ICU stays, and fewer deaths. And we commissioned a firm, RTI Health Solutions, to perform an economic study on what it would save the medical system in the US if you diagnose sepsis a lot earlier. And the cost savings are enormous, $3 to $3.9 million per hospital. And because there's a lot of hospitals in the US, that's $16 to $22 billion annually. So this has enormous potential. Our therapeutic technology is a proprietary peptide technology, and this directly addresses the ineffectiveness of current treatment options by suppressing biofilm formation and reducing the main pathology associated with biofilms, which is inflammation. So you can see here a human skin that we're able to grow in the lab, and then we apply bacteria to it, such as this super pathogen MRSA, which is a very resistant organism. And this forms a biofilm on the surface of human skin. And I think you can see that it really destroys the surface of the skin compared to the figure on the right-hand side that you're looking at. So these purple clusters are actually mounds of bacteria on the surface of the skin. And these mounds of bacteria are highly resistant adapted communities that grow on body surfaces. So they're very resistant to antibiotic treatment. So our peptide that we've developed destroys the biofilm and restores the skin back to its health, as you can see on the right-hand side. So this is a huge, urgent, unmet medical need. We know that there are 2.8 million genetically antibiotic-resistant infections in the US annually. This is called AMR. This contributes to a lot of deaths. But in the biofilm situation, there are even greater resistance to antibiotics, and there are no approved treatments. We also know that there are 11 million deaths globally from sepsis because antibiotic treatment often fails in sepsis patients. 22% of the time, people die. And the most dangerous pathogens, which are called the escape pathogens, include untreatable variants. So MRSA, just as one example of one of these escape pathogens, contributes to more deaths in the USA per year than homicide, Parkinson's, emphysema, and HIV/AIDS combined. This is a huge issue. So highlights of our peptide technology: we have strong antibiofilm activity that Dr. Haney will show you. They work in combination with conventional antibiotics to overcome antibiotic resistance. We've been able to demonstrate that they're safe and effective in animal model infections of real biofilm infections, including sinusitis, abscesses, wounds, and oral infections. They have strong anti-inflammatory activity in vivo that works as well as the non-steroidal anti-inflammatory drug indomethacin, highly used in medicine. They also can modulate the immune system to suppress inflammation, but boost protective immunity. And they have the combined activities, so you can come up with peptides that have multiple different activities. So just to mention a little bit of introduction to bacterial biofilms. So biofilms are communities of bacteria, and you can see one such on the right-hand side there. These are often attached to surfaces. So it's an adaptive growth state of bacteria. So the bacteria actually adapt to a stressful situation, and they adapt by sitting down on the surface and forming these mounds of bacteria that are held together by a protective coat called a matrix that is made up of polysaccharide. And you can see the wispy material in between the round cells that holds this biofilm together. Some consider this to be the natural growth state of bacteria. It certainly is in infections because it's 2/3 of all infections. The estimated cumulative economic significance of biofilms is $5 trillion a year. That's a lot. So this gives you an illustration of some of the biofilm infections. There are both device-related infections that we'll talk about right towards the end of the presentation, and tissue-related infections. And you can see that the tissue-related infections cover a range of different areas of treatment, including eye infections, ear infections, lung infections, endocarditis, urinary tract infections, and oral biofilm infections. So as I mentioned, these are very resistant to conventional antibiotics. They're clinically relevant because they're 65% of all bacterial infections. The estimated cost of global healthcare is $368 billion annually. And there are no biofilm-specific therapies approved for clinical use. And as I mentioned, there are many, many specific and discrete opportunities for addressing large unsatisfied markets. So I'm going to turn it over to Dr. Haney, who's going to give you a little bit of an idea of where we're progressing scientifically. Over to you, Evan.
All right, thanks very much, Bob. So Chris mentioned at the start that we're going to go through some complicated science and data today, but I'm going to try and keep it simple for everybody. So as Bob mentioned, I'm going to give you kind of a quick snapshot of some of the technologies that we're developing. I'm going to start with our diagnostic technology, and then we'll talk a little bit about the peptide therapeutics. So next slide, please, Bob. Perfect, this way. So our diagnostic test, we are calling it Sepset ER. And as Bob mentioned, we're trying to target patients to be able to diagnose them when they enter into the emergency room. It's a molecular diagnostic test that uses reverse transcriptase qPCR, a reaction, which is the same technology that's been used for diagnosing COVID in PCR tests. So when you hear of a COVID PCR test, it's the same as what we're using here. We are focusing on a patient-specific immune response that is actually being able to diagnose those patients who are going on to develop sepsis. This immune response, this gene signature, we find has greater sensitivity and specificity compared to things that are currently used in the hospital. And it allows an earlier recognition of sepsis at a time when patients are admitted to the emergency room. So as Bob alluded to, being able to identify patients early can hopefully translate to improved survival and better patient outcomes. So the table at the bottom just shows you some performance statistics that we've been able to identify using some of our data sets and publicly published data sets. We find that our gene signature is highly accurate when we look at patient samples that are collected from the emergency room. We have an accuracy of 92%, and this means that we're able to identify, this is a combination of a sensitivity, which is the ability to correctly diagnose somebody with sepsis. So we have about an 88% sensitivity. And then we also have a 94% specificity. And so specificity is also important because this is the ability to correctly identify those patients who do not have sepsis. So we find a very good balance between our sensitivity and specificity. We can see in other data sets that we've collected, so using a PCR reaction, not in our test, but using our underlying algorithm, we find that the test also performs very well. So this is basically the same technology, and we can see that our gene signature is, again, very good at separating patients according to their sepsis status or not. They're using a published data set, so that's the Arunchalam paper in 2020. We find again this is from a transcriptomics study, we see again that our gene expression signature works very well. And we have a separate data cohort collected from patients in Quebec, and we see again very similar sensitivity and specificity. So we see that this gene expression signature seems to be robust and is holding up across multiple different patient populations. The other interesting thing is that we find that this is very specific for sepsis patients. So you see they're highlighted in red. gene expression signature from cancer patients. And you can see here that the accuracy is about 50%, which you would expect because cancer patients don't necessarily have sepsis. So our gene signature is not picking up cancer, it's actually picking up sepsis from patients. And so that's reflected in the very low sensitivity. So none of these patients have sepsis. And so you would expect to see a low number there. And then same thing for the specificity, you see that it's correctly identifying the patients as not having sepsis. And then in green at the bottom is just a couple of other published or currently available sepsis diagnostic tests on the market. So this is using our data and what we are trying to do for emergency room patients, but using their gene signatures. And you can see that the accuracy and sensitivity and specificity is lower. So it seems like for emergency room patients, for what we're trying to actually diagnose, our signature works very well. So next slide, please, Bob. So not to get into too much, but this is just to give you a little bit of a taste of what happens with the SEPSET ER test. On the left side there, you see what's called an amplification plot. And this is actually the basically raw data that comes off the RT-qPCR machine. And you can see each one of these lines on the graph is corresponding to one of our signature genes of interest or one of two housekeeping genes that we have. And where that line goes up and crosses over a certain threshold value, that is directly proportional to the number of copies of the gene of interest that we have in the cell, or in our sample, sorry. And what we can do is we can take all of these values and we collect it as raw data from the instrument, and then we just upload that into our software or our algorithm that actually sorts patients on whether or not they have sepsis or not. And so we use this as an input for our algorithm. This algorithm then predicts the patient sepsis, so whether or not they're going to go on to severe sepsis or not. And then it just spits out a sepsis diagnostic score to inform the treatment patients for the doctor. So it's very simple to implement and requires very little manipulation or anything like that from the technician who will eventually run it. So just as a last note for the diagnostic side, we've done that sample is actually on a healthy patient sample, and we're just in the process of being able to get some sepsis patient samples to ensure that the diagnostic works well in sepsis patients. So moving on to our peptide technology. So as Bob mentioned, this is a technology that looks at biofilm-associated infections. And he already showed you this slide, this image to be able that you can see biofilms, for example, growing on skin surface. You can see that the peptides kill the biofilms and can reverse skin damage. So next slide, please. I'm just going to give you a quick snapshot or a quick overview of sort of our philosophy or how we come up with identifying new peptides. So we really rely on peptide libraries, and we do it with something called spot synthesized peptide arrays. This allows us to be able to look at a large number of peptide sequences and also expand beyond naturally occurring amino acids. So it really gives us some versatility and flexibility in the peptide sequences that we're able to access. So from a peptide library, we'll get those synthesized and then we'll screen them for some sort of activity. Most of the time we're looking at anti-biofilm activity. And then from that, we'll take the top hits that we identify in our screen. So we're taking the most active peptides, we'll make them to high purity, and then we'll validate them in some of the models of biofilm-associated infections. And I'm going to show you data from some of these more sophisticated biofilm infections. But the other nice thing about the way that we do this is that we can also iterate on this peptide design. So we can take the results from these screens and we can feed them back in, either making new peptide libraries, or we can do some machine learning and computational modeling to be able to come up with new peptides that have enhanced activity and really keep our product pipeline full. So now next slide, please, Bob. So the first target that we're looking at is oral biofilm. So oral plaque is actually a polymicrobial biofilm that forms around your teeth and in the oral cavity. So this is typically removed mechanically. So whenever you go to the dentist and they'll do what they call scaling, that's actually them kind of pulling away and pulling off this biofilm that's attached to your teeth. It's also associated with dentures and dental implants. But this microbial community can contribute to infections in the oral cavity, and these really cannot be treated with antibiotics. So they are resistant to the effects of antibiotics. And just as a little aside, the global market for the oral healthcare is very large. As you can imagine, it's estimated to be in the neighborhood of $47 billion. So there's a tremendous market potential here. Next slide, please, Bob. So what we've found is that our peptides can actually work very well against oral biofilms. And this is just showing you some data of biofilms grown on hydroxyapatite. And it's just a material that can serve as a surrogate for like a teeth surface. And so over on the left there, you can see looking at these biofilms grown on this hydroxyapatite using a technique called confocal microscopy. So for this, we use some fluorescent dyes, and you don't need to know the whole amount of it, but basically if you see green, that's a living biofilm, and if you see red, that's a dead biofilm. So you can quite clearly see that without peptide, you can see that the biofilm is very healthy, all the cells are green, whereas when you treat with our peptide, in this case, this is one that's published, it's called 1018, you can see that most of the biofilm cells are dead. So these peptides really work well on the biofilms that are growing on this hydroxyapatite. And I should point out that these are actually biofilms derived from donor saliva. So these are fairly close to natural plaque that you would have in your mouth. They also can synergize with other solutions that are used as oral mouthwashes. So that's either combined with chlorhexidine or EDTA. And you can see that the combination of the two works very well, and you still see the dead biofilms. They also work on dentin biofilms. So these are kind of biofilms that are growing within the tooth surface. And you can see that they can penetrate into this dentin and actually kill the biofilms as well. And you can just see how the peptide treatment affects the biofilm surface as well. So that's using the scanning electron microscopy on the right there. You can see that the biofilm sort of has a lot of different matrix components and cells combined in there. And then when you treat with peptide, you can clearly see that the structure changes and you're really altering the structure of the biofilm. We've got a few slides from now. You'll actually get really close in and see what the effect is on the cell surface. So next slide, please. One more back. So we've, in collaboration with our collaborators at UBC, they've actually been able to take this up into human volunteers. So this is actually taking a hydroxyapatite disc and basically putting it in somebody's mouth for up to 5 days. And so for the first 3 days, you just leave that hydroxyapatite in that person's mouth, and it allows a biofilm to form. And you can see that in the top panel there. But then after that, you then start to treat these with a solution or just a mouthwash rinse that incorporates our peptide. And you can see over on the right-hand side there, after day 3, when the mouthwash starts to occur, you can now start to see the biofilm cells start to die. Whereas when you treat it with just water, even lower concentrations of the peptide alone or the chlorhexidine, they're not as effective. So this really demonstrates the potential of the peptides to be able to combine with already available oral mouthwashes and really be able to go after some of those biofilms. All right, next slide, please, Bob. So the next one I'm going to talk to you about is infected wounds. So wound infections are very common, and we've particularly been looking at them in their issues in war zones. And the reason that can happen is that it's very easy for any wound to become contaminated by bacteria and cause biofilms to form, and this can really prevent healing. In addition to that, there's potential for uses in surgical dressing. So obviously that would be another type of wound that we could look at. And we're looking at incorporating our peptide technologies into a wound dressing. And this allows us to look at them more as medical devices and not a therapeutic. So this is in collaboration with a company out of Ithaca named iFiber. And we've been working with them and have received some US Department of Defense funding to be able to incorporate peptides into wound dressings. And so next slide, please. We'll show you a little bit of some of the activity of these peptides. So this is one of the assays that we often use, is growing some skin. It's called an organoid model of skin. So it's not quite human skin, but it's using cells derived from skin to then be able to generate this stratified skin layer. So that you can see where it's being highlighted there. You can see that you get this structure of skin that we grow in a dish. We can then infect the surface of that with biofilms, and you can see that highlighted in the middle there. And then when we treat with peptide, you can see that the biofilm goes away. So over on the right is another little trick that we use. We can actually tag the bacteria to emit light. And so this allows us to be able to visualize the biofilm growing on the skin surface before we treat it. So you can see that that's that top panel. And then now you can see that after we treat with peptide, you can see that just treating it with water, the light persists. And then when you treat it with the peptide, all that light goes away. And that's quantified over on the right there, where you can now see that peptide treatment, you see a big reduction in the number of cells that you get on the surface. So I promised you a few slides ago, we were going to get really up close to these bacterial cells. So you can now see that on the right, you see the untreated Pseudomonas biofilm growing on that skin surface. And then obviously you can see a very stark difference when you actually treat it with the peptide. The cells develop this furry structure. We know that the cells are doing some really amazing things to the cells and actually causing them to die. And the other thing that we'll just highlight here is that this system also allows us to pick up, so they also work better than standard treatments. So these are things like fusidic acid or mupirocin or silver nitrate. So these are approved things on the market that actually still don't work very well, mainly because of this biofilm state. So our peptides appear to work better than a lot of these. In addition, this system also allows us to look at the anti-inflammatory effects of the peptide. And so that's just kind of quantified over there on the right. And you can see that treatment with peptide, this is looking at the expression of an inflammatory marker called interleukin-6. And you can see that compared to the infected cells, the MRSA, you can see a reduction in the production of IL-6, suggesting that the peptide is inducing an anti-inflammatory effect. Next slide. So these are some of the new peptides that we've come up with. So we can see now that you get, when we look at the light emitted, that the light goes away upon peptide treatment. We can then also look at that quantified. We get really great activity against both MRSA and against Pseudomonas. And when we look at them under the microscope again, we really see some very striking effects of the peptide on the bacterial surface. So you get kind of a very rough surface that emerges, and there's clearly effects of the peptide happening on these biofilm cells. And then lastly, just the peptides being able to synergize with antibiotics. So this is the next thing that we're looking at, is trying to combine the effects of the peptide with an antibiotic to be able to enhance the activity of both. And so shown over on the left is some work that we've been doing combining peptides with an antibiotic. And you can see that on their own, if you look at the effect of the antibiotic alone, this is ABX3. And with any of these different peptides, you can see that the effect is decent. But then when you combine them together, you actually see an enhancement in the bacterial killing on the skin surface. And in addition, you also see that you have very little bacteria that gets recovered from the dressing itself. And so you can see even one of them, you get completely sterilization of the dressing itself, which is important when you think about these in terms of medical devices. And then over on the right, this is also showing this combined effect, this synergy effect in a pig model of biofilm infection. So pigs have actually the most similar type of skin to humans. And you can now see that the combination of a peptide, in this case with ciprofloxacin, you can get almost a 3-log reduction for Staph aureus, whereas you get almost a 4-log reduction when you're looking at a Pseudomonas infection. So these also work really well in a very similar biofilm infection model to humans. All right, next slide. And then the last one that we're really excited to be going after is something called chronic rhinosinusitis. So this is a very common bacterial biofilm infection. Affects 1 to 5% of the population, causes 4 million doctor visits per year. And the most common treatment for this is actually surgical removal of the infected tissue. So the doctor will actually go in there and scrape out the sinus cavity, which, as I'm sure you can all imagine, it's not very pleasant. Antibiotics do not work because of the biofilm. And the other treatments only treat the symptoms of it, not the cause of it. So not the cause of this biofilm. So there are approved monoclonal antibody drugs, but these are very expensive. They cost $10,000 to $50,000 per year, and they do help in some situations, but the evidence of efficacy is still, in the range of 35% of patients. So our peptides, what we've been able to look at is the effect of the peptides in a bacterial sinusitis model in mice. And so that's showing down here on the right. This, again, we use these light emitting bacteria to be able to visualize the biofilm growing in the mouse nasal cavity. And you can see now that when you image it 5 days post-infection, you have a very high density of bacteria in the nasal cavity of the mice. But when you treat it with the peptide, you can clearly see that light has gone away. And that's demonstrating the effectiveness of our peptide of killing those bacterial cells. And that's, again, just quantified over there on the right. You can see that we have several peptides that can all reduce the bacterial burden in the nasal cavity of the mice. And again, we also see that this is accompanied by an anti-inflammatory response upon treatment with the peptides. And so this is my last slide here, is looking at some new peptides that we've got. If you go to the next one, Bob. And so now we've got a few new peptides that we've identified that actually have this, and we have an enhanced antibacterial effect compared to DJK5, which is one that we published on. And so now we're very excited to see and move these ones forward to actually be able to get these ones into some preclinical studies. And so I think that's it for me giving an overview of some of the technology. And I'm going to pass it over to our COO, Tim Murphy, who's going to give you an update on some of our corporate update.
All right, thanks very much, Evan. Thanks, Bob. Before I talk about the management and the structure of the board and everything, I'd like to just describe something we don't have a slide for, but we probably should, which is the structure of the company itself. So Asep Medical Holdings, Inc. is a holding company, and it has 3 portfolio companies, the first of which is Sepset Biosciences, Inc., which is the one that's developing the Sepset ER test, which Bob and Evan both mentioned. So that's a diagnostic test for sepsis. The second major portfolio company is ABT Innovations, Inc., and that's where all the peptide research is coming out of, and primarily based in the Hancock lab at University of British Columbia. And then most recently, we just acquired a company called Safecoat Medical, Inc., which we'll talk about a little bit more in the presentation. Next slide, Bob. Okay, so our management team is headed up by Dr. Robert Hancock, whom you've all met. He's our chairman and our chief executive officer. For business development, our chief biz dev officer is Dr. Fadia Sad. She brings a lot of experience to the table in these types of joint ventures and deals, which we'll also talk about a little bit later in further slides. I'm acting as the chief operating officer, primarily to keep things moving on a number of fronts within the company. And Jacqueline Tucker is our chief financial officer. And so she brings a lot of experience at both the public and private level in terms of governance, and has been a really great addition to our team. And we've got Dr. Evan Haney, who you met, who is our chief science officer, who is assisting in the lab quite a bit, but also doing a lot of the regulatory approval work, which we'll touch on in the next slides. Next slide, please, Bob. No, I think we need one more. I think one back. We're missing our board of directors. There we go. Okay, so the board's comprised of 4 members presently. So we've got Dr. Hancock, who you met, Daryl Norgard, who's a member of our audit committee and also got a deep financial background, myself from a legal and operational standpoint. And we most recently added Dr. Richard Heinzel, who is one of the founders of Doctors Without Borders Canada and a globally renowned physician and entrepreneur, and he's been a great addition to our board of directors. Okay, next slide, please. All right, so generally speaking, our corporate strategy is to maximize shareholder value, but how do we do that? Well, Asep is really, and its subsidiary companies are about researching and developing intellectual property. Our only valuable asset is our intellectual property, generally speaking. And so we get that intellectual property, we take it out to the world, and we develop partnerships with larger entities in order to help commercialize the IP. So as you've heard, there's a number of novel strategies. The way that the company is approaching dealing with infectious disease is quite novel in the marketplace. There's been no drugs approved for biofilm, as we've seen, and yet it's a major focus of the work that we do. In terms of how we get that out there, in Sepset, the Sepset ER test obviously requires a regulatory approval. So we've developed partnerships in order to get that approved through the FDA. We're in that regulatory pipeline, and then we're working with partners to get it approved in other jurisdictions. Okay, next slide, please. Here we go. So I won't talk too much about this one, except to say that you can see in the yellow sections, those are basically applications coming through Sepset, which is one of our portfolio companies. And in the green, you can see that's peptide-based technology, which is coming out of ABT. So Sepset ER is still an early stage technology. It has not been approved by the FDA, but we are working towards approval. And on the ABT side, same thing. We haven't received any approvals at a regulatory level. However, we're at a various stage of clinical trials and early stages of regulatory approval for a number of the applications. Next slide, please. So partnerships are really the lifeblood of how we're developing this company. We've already made a couple announcements, which are quite exciting, related to Sepset ER. Most recently, we just announced a partnership with Seaspring, which is a large company based out of the Principate of Bahrain. And actually, Seaspring is backed by the Prince of Bahrain himself, and that's a partnership for the Middle East and North Africa for the exploitation of the Sepset ER test in those territories. And we have a lot of hopes about that one. So we signed a term sheet for that, and we are in the process of finalizing our definitive joint venture agreement with them. We've also got some interesting prospects in the markets of China. We can't say specifically who yet, because we haven't put out an announcement about it yet, but we hope to soon announce a partnership in China with a major Chinese partner to exploit our Sepset ER test in that jurisdiction. On the diagnostic supply and technical side, we work with Thermo Fisher, which is a major diagnostics company. The dental mouthwash, which Evan described earlier, there was some human trials that were done at UBC with Dr. Ya Shen. However, we've now expanded that through a partnership with Bohai Biomedical in China. So there's going to be a 4,000 person test in China, which is going to determine feasibility in that market. And we hope that that's going to turn into a joint venture as well. Evan also mentioned the wound healing partnership with iFiber and its relationship with the Department of Defense in the United States. So that's continuing to come along. We hope to make more announcements about that as it develops. And we've also got a formulation partnership with the Canadian Nanomedicines Innovation. So next slide, please. Actually, I should say, the next slide, we talk about the non-dilutive funding. So let's do that. So we're very blessed in this company to have direct access to the Hancock Lab. The Hancock Lab has been around for decades now, and it is a leading lab globally for infectious diseases. A number of the technologies that are in the companies have come out of it. a matter of fact, I think they all have, except, some in partnership with some other labs. So we save quite a bit of money by having a relationship with the Hancock lab. We don't have to go out and buy all the equipment, directly hire all the technicians. We're able to leverage the relationship with the Hancock lab in order to reduce our overhead significantly. So in the industry standard, you'll see here it's 40% versus 200%. So it's a major savings. Similarly, through our regulatory process, as we get things approved, we're going to be partnering with contract research organizations. So these are the experts in regulatory approval. IQVIA is a great example whom we've worked with on Sepset ER, and I expect that we'll continue to work with as we guide Sepset ER through FDA approval. We've also got some key internal hires recently. Well, some recently, some not so recently. We've got Dr. Peter Zhang, who's been helping us on machine learning and artificial intelligence. Artificial intelligence is a bit of a buzzword these days in the markets, but we do utilize artificial intelligence and machine learning in the Sepset ER diagnostic. So in other words, there is a software layer in the test itself, which analyzes the results from the peptides, which enables us to get more accurate results. And then we've also got Sheila Galan, who's come in as an expert in quality assurance and regulatory affairs with Asep. And so this is extremely important as we go through approval with the FDA and other regulatory associations globally. Quality assurance and QMS is essential in order for us to get those approvals. Next slide, please. Okay, non-dilutive grant funding. So we've raised millions and millions of dollars in non-dilutive funding over the last decade. The technology that was presented by Dr. Hancock and Dr. Haney, it didn't just arrive at our doorstep yesterday. This has been developed over many years, and through the assistance of many important partnerships with NGOs, with government, with educational institutions, who have helped us finance and develop this technology. So although we only went public a little over a year ago, this company has really been, a decade in the making. Next slide, please. So most recently, we added to the Asep portfolio through the acquisition of Safecoat Medical Inc., which is an extremely interesting technology for preventing device-related infections. Essentially, I don't want to go too much into the science because I'm not the science guy. We've got other people in the group that do that. But it is an anti-fouling technology, which essentially adheres to medical devices, for example, a catheter, and allows us to apply the peptide to it so that you can see at the bottom there, there's near the substrate, it looks like a bit of a Christmas tree. So you can see how the peptides will adhere to the device or the instrument and actually prevent the infection from furthering. This technology has been proven in principle with animal subjects and animal models, and we will continue to develop that. We think it's going to be a terrific market for us, and it was a great opportunity for us to be able to bring that into Asep and add it to our existing portfolio. And there is quite a bit of overlap, obviously, between ABT, which does peptide work, and Safecoat, which is an anti-fouling technology which uses peptides. Next slide, please. Okay, I'll hand over to Bob to close this off, and then I think we'll open it up for a few questions.
Okay, so I hope that people have taken a lot from this presentation today. Our company is basically dedicated to dealing with this issue of antibiotic failure, and I think we have some unique and potentially very fruitful approaches. We've managed to, in a sense, find a niche in something that is not a niche market. It's really a set of massive markets but we found something that we can do better than anybody else. And I think that speaks well for shareholders. So I'm going to stop sharing now, and we're happy to answer any questions which you can, and Chris will explain to you how to do that.
Thanks, guys. Thank you, everybody. Great presentation. Ladies and gentlemen, there will not be a test at the end of this webinar, so you don't have to worry about that. I do have a question here from one of our investors, and he would like to know if we could get a bit more detail on the progress made towards the 501(k) application, what the timeline might be. Evan, are you able to answer that?
So I can give a little bit, I guess. So it's a 510K. I think all of us, we're still trying to wrap our brains around that. Maybe we keep thinking of 401K, but it's a 510K application. And so it's for us to be able to demonstrate substantial equivalency to some device that's on the market. So we're in the process of working towards that. There's a fair number of steps that are involved with that along the way. And so we are in the process of developing our clinical study protocol. So that's using one of our CROs that was mentioned, IQVIA. They're helping us develop our clinical study protocol, and also to get together a documentation that we need to have a pre-submission meeting with the FDA. So we're hoping to do that in the near future. And once we have that pre-submission meeting with the FDA, we'll be able to discuss with them the plans for our clinical study and to ask them a few questions to make sure that, what we intend to do would be, that they think that that is a viable path forward. Once we have that, then we would initiate our clinical study. This would be a clinical study that's going to be run at least 3 sites. We're looking to do a couple of sites in the US and probably 1 site in Canada, but we don't anticipate that our clinical study is going to take very long to perform. So the thing with sepsis, it's actually a very common condition, right? So a lot of any emergency department in a large center is going to have a decent number of patients that are going to come in every day that a doctor will suspect of sepsis. And so, we think that we would be able to get fairly quick enrollment and be able to assess the test very quickly by identifying a large enough patient population in that. Once we have that clinical study done, so the clinical study will depend on how fast we can get patients recruited and being able to have our sites to be able to do that. But once we have that done, then we'll be able to put together all of our final paperwork and then submit that to the FDA for 510K. We're looking to get what's called 510K clearance, which allows us to actually sell our test and market it. So that gives you an overview of kind of the steps involved. Some of those things, once we actually submit paperwork, it's 3 months once we submit things to get a pre-submission meeting. And I think it's about a similar timeline once you actually submit your final paperwork. It'll take a few months for the FDA to review, and then you have a final meeting. So really a lot of it hinges on that, being able to get that clinical study done in a timely manner, but that gives you an idea of the timeline that would be involved.
Thanks, Evan. Good stuff.
Sorry, so just to add to that, it's a little bit different from therapeutic studies where you have to do phase 1, phase 2, and phase 3. In this case, the 510K process is a single clinical study. It usually involves, or we're advised by our contract research organization, it usually involves about 600 to 800 patients. That is not a huge number. And it's a lot cheaper than doing a therapeutic trial because you don't have to pay for all of the clinical stuff. You really, literally, the test involves taking a blood sample and then sticking it into your diagnostic machine. That then feeds the results directly into an algorithm that then gives the doctor advice as to what the likelihood is that the patient will have severe sepsis. So those individuals who are advised that the doctor is having severe sepsis, the doctor would initiate immediate goal-oriented therapy. For those patients who don't, the physician could perhaps avoid putting that patient in the ICU and also avoid the use of expensive antibiotics, thus decreasing antimicrobial resistance. So there's actually benefit to both a positive diagnosis and a negative diagnosis of sepsis. Just speaking personally, my goal is that we'll get through most of this process this year, trying for approval by the end of the year, but not everything is in our control. The FDA may insist that we gather more information after we filed our approval. They may also insist that we design our trial a little bit differently. So that's the sort of the basis for the pre 510K meeting with the FDA, is to try to get that sort of advice to make sure that we can do it as expeditiously as possible. Nevertheless, in biotech, this represents really a near-term goal, because for therapeutics, as I'm sure you're aware, usually the goal is to make money in more like 5 to 8 years.
Yeah, it's also worth mentioning, Bob, maybe you can expand on this. Some of the technologies that we have don't even require 510K approval.
510K.
Yeah, so nothing is absolutely certain in the regulatory world, but the mouthwash application is usually treated under what's called cosmetics legislation. And so what we're looking for is a high-end mouthwash, the sort of thing that you would go into your dental office and you would immediately be given this mouthwash. Currently, I looked the other day when I went to the dentist, they use xylitol solutions, which are pretty well useless. And this would then prepare the patient by decreasing the biofilm growth in their mouth. So that would be our initial attempt at a product. And I think that can be quite fast-tracked, because it doesn't have to go through the regulatory process. And then Evan also mentioned the wound healing area, which is not done under therapeutic legislation, but under device legislation. So the bandage plus the treatment is considered to be a device. And then that can also be relatively fast-tracked. So we're very hopeful that we'll be able to get approval sometime next year.
Good stuff, guys. Thank you. Another question here is, when do we expect to need future financing and how much? The second part of the question is, do we expect future analyst coverage to help support share price? Tim, maybe you could help with that.
Yeah, I mean, we're thinking about, I mean, there's a number of different ways that we get funding, right? Some of it is the non-dilutive funding that's been mentioned so far. So there's, a continual flurry of grant applications and things that are going out for the various companies and technologies. So that's something which I think reduces the burden for shareholders in terms of allowing us to continue to research and develop our intellectual property. Presently, I won't speak too much to this, but our cash situation, from my perspective, is adequate for what we're trying to do. That being said, we are always thinking about new ways to raise money. I don't want to get too ahead of some of the announcements that we're going to make over the next few weeks. I do expect that we will be doing a financing at some point in the next few months. I can't say exactly how much. That's going to depend on the board getting together and discussing it. But I do believe that we will be doing a financing next few months, and that will primarily be designed in order to help us get more products into the regulatory pipeline for approval. In terms of analyst coverage, we haven't really gone out and sort of banged on the doors of Bay Street and Wall Street yet. However, we have recently started some fairly significant discussions with a major US investment bank. I can't say much about it right now because nothing has been announced, but those discussions are in progress. And in terms of analyst coverage here in Canada, we are actually going to have some meetings scheduled over the next couple months with analysts in Toronto, and in order to get them to cover our stock, which we think is one of the most exciting stocks in Canada at the moment.
So I just want to add to this. We have enough money on hand to go for our near-term targets, which, as we mentioned, is the sepsis ER diagnostic test, plus the mouthwash. So we have enough money on hand to do that part of it. However, we're very ambitious, and we wish to achieve some of our other goals, because all of them have huge money spending potential. One thing I would add, over and above money raised in the marketplace and money raised through non-dilutive funding or grant proposals, we also have a tremendous opportunity through partnership. And the idea is that as we move along, because we're developing such a large range of different technology ideas, that we will pick up partners who will take over the really the expenses for the particular technology.
Yeah, that's a good point, Bob. I mean, some of the partnerships that we've been negotiating right now with the current partners definitely includes a revenue stream for Asep and its portfolio companies.
Yep.
Okay, I've got one more question here, which I think Tim can probably speak to as well, and that is, can you speak to the idea or the issue around why the stock fluctuates so much in comparison to other stocks? And I think this has a lot to do with the fact we're in a bull market. And
More bearish than bullish, I would say.
Sorry, in a bear market. But and I think we've seen some Canadian stock owners selling. Tim, can you expand on that a bit?
Yeah, I mean, Asep went public in November of 2021, which was right about the time that the market started to get into bear territory. So I think essentially for the entire time that we've been a public company, we've been in a bear market. That being said, I think that our stock has been quite resilient. We've more than doubled the stock price in the last 3 months. We're continuing to get our good news out into the market, and there are many, many good things happening in the company. And as you've seen recently, we've signed some important term sheets. We've got the feasibility with Bohai. We've got some other major announcements which we look forward to sharing over the coming weeks and months. All of these things do affect people's when they buy or when they sell. Those decisions of people to buy or sell, of course, are personal, and we can't control that. We have recently engaged in the expansion into the European market in order to reach investors on that side of the pond through an engagement with Bullvestor to start to tell our story in Europe, which I think has been quite successful recently. We've invited a whole new group of shareholders from Europe into the company. And that program is going to continue to run, and we're going to continue to get our good news out there. And we hope that it'll result in a good return for all of our shareholders.
Yes, in fact, we actually have 5 attendees today who are from Germany, so welcome them.
Yeah, I think speaking for the company, I think that one thing that's pleased me greatly is that we've had a lot of very loyal investors who are sticking with us, and I think they understand that a company such as ours is not going to be an instant overnight success. It's not like selling Coca-Cola or something like that. There has to be a little bit of patience. And I think generally speaking, the fact that the selling has been mostly done a fairly low volume basically speaks to the fact that people do believe in the long-term future of this company, which I truly believe in.
Okay, we're at 11:07. I think we have one more time for one more question. I'm not sure who would like to answer this, maybe you, Bob, but could you summarize quickly why you think the company has large potential upside?
Yes, so I've started a number of companies today, and virtually all of them worried about money rather than direction. So the whole idea was to make a couple of bucks for every buck that went in. That's essentially the venture capital route. Our company basically is looking to the future. We genuinely want to produce solutions for unmet medical needs. So I think our large upside is based on the fact that we're dealing with unique products that have huge potential in very large marketplaces. So we told you about the sepsis diagnostic market, which is definitely unsatisfied. And we told you about the biofilm market, which is definitely unsatisfied, with no products actually developed to date for that particular marketplace. So these are tremendously large markets and I think our approaches are quite novel. And if you look around, you won't see many companies addressing these particular issues. So I think our huge upside is that. And secondly, because we'll do anything to progress our products. And I think the thing that we've done best to progress our products is to partner when it's necessary, rather than try to be an island unto ourselves. We're trying to bring in partners to really grow this company appropriately.
Thanks, Bob. Maybe I'll just add a little bit to that, if I may, from my perspective. I mean, it's a relatively early stage company in terms of, seeing the public seeing ASEP. That being said, though, there's a lot of expertise in this company, as I'm sure you saw throughout the course of this webinar. The confidence level within the team is quite high regarding regulatory approval. And from a regulatory standpoint, we're still somewhat early stage. However, once regulatory approval is achieved for one or more of the products and technologies that we're developing, the net asset value of this company is going to substantially increase. And some of these partners that Bob was talking about, these are some of the largest pharma companies and organizations in the world, and in some cases, and they are taking our call. Like, they are very interested in what we have, and they recognize the expertise in the team. They recognize the quality of the research, and despite the fact that we haven't achieved regulatory approval till this point, they want to start talking with us. So we've had a lot of good discussions with them, and from my perspective, the future of this company is incredibly bright.
Good. Thank you very much to all of our attendees today. Thank you to our panelists. This concludes our webinar for today. You all got my email address. Should you have any further questions, I'm very happy to take any emails, get things answers from our investors and prospects alike. And once again, thank you for attending, and we'll make sure you know about our next webinar planned within the next couple of months. Thanks, everybody.
Thank you.
Thank you.